Or you...
Okay. Yeah. Okay. So then, when did they measure corticosterone, and how did they collect the blood? So, they just euthanized the mice with CO2 inhalation, and then they did ELISA for the corticosterone.
Oh, so they euthanized them.
Yeah.
So this was the terminal blood.
Mm.
Okay, and at what time was it? In the morning or in the evening? That was my question. Sorry, I was not specific.
I think it doesn't say. No, just fasted. No, maybe it doesn't say the time.
They were fasted for 6 hours prior to blood collection. That's for plasma insulin measurements.
Oh yeah, sorry.
That is for they use GGT and IgG, as I remember. That's a different thing. But for corticosterone, they don't say, right?
No.
Did you see the levels of the corticosterone that they have? Somewhat different than what we got. That's why I was yeah, actually, it's here. That's why I was interested whether it was done in the morning or in the evening. Which, as you know, like in the evening, like late evening,
it's lower.
They have higher. We have lower.
And they okay, there.
So
yes.
They are getting more active just before waking up. So just before, if it is like cycle until 7:30 in the evening, so then just several, maybe 1 hour before, the corticosterone surges to let them wake up and be fit. Yeah? While in the morning, it's kind of low. So,
but as far as they're euthanized and the blood is collected and measured at the same time, both control and experiment.
Sure, still, I mean, this is great. You have to have it at the same time, but the question is, which exactly corticosterone values are we looking at right now?
Okay, then. Oh, then you asked me a lot of questions about SIM1 expression in mouse embryogenesis and things like that. So, if there is like differential expression during the embryonic state or the adult state, and then I searched a little bit. It was a quick search, I have to say, in the protein atlas.
That's their job to discuss that. Didn't they mention that
No.
in the introduction or in the discussion?
No. Embryonic word is only mentioned once. And it's not related to this.
So why I was asking? Because they don't use ERT2 line, but they use CRE line. So all the phenotypes that they are looking at are embryonic, strictly speaking. Those genes start to be expressed in the embryonic stage. They don't study here the adult phenotype, or at least strict only adult phenotype. It might be that this effect is caused by the adult stage and there is no influence on the embryonic stage at all. Could be, but it is hard to believe. I would probably suggest that the embryonic involvement of the loss of miR-7 and MicroRNAs are really critical for the embryonic development. So during the development, MicroRNAs do really a lot of different functions in order to make this differentiation, to all this interplay of different lineages and so on and so on. And this is specifically true for MicroRNAs, and specifically true for those which are highly abundant, like miR-7. So all the phenotypes which are described in this paper are embryonic, primarily. And of course, they continue to express in the adulthood. Of course, they also have some impact on the adult stage, probably, But as I see from this paper, like they don't really discuss that. So it means that they are claiming, okay, that this is the function in how it functions in different regions. Not really. I would say this is how this microRNA does impact the those lineages of the SIM1 genes which start to express in the embryonic development, and how it shapes their further development differentiation. So it might be that just there were not enough neurons formed from this lineage because miR-7 was absent, and this we can also see there. So what they say when they show that there are less neurons. Remember?
Yeah, I have a question about that.
This is not because they are dying, most probably, but it is because not enough of them were formed, because miR-7 profoundly changes the these precursors of the neurons in a way that they didn't really form enough of those. So the phenotype is embryonic. This is at least what I would say. Yeah. So primarily, maybe there is some also contribution from the adult function, but for me it looks like really embryonic. Moreover, it could be that some of those SIM1 genes during embryonic stages, they stop to express in those neurons, and they change the identity towards different cell type, which is true for some other.
What would that happen? Because of the Cre presence?
No. The Cre will be expressed there anyway, and we have the knockout anyway because there is flux gene, so those cells will have will be fluxed in those neuronal lineages. But later, those SIM1 neurons might switch off the SIM1 promoter, so they stop to be SIM1
because
neurons.
it's not functional, so why to express it?
No, it might be that it is important to be expressed during the embryogenesis, but it is not important during the adulthood. That's why they might switch. Like CAM-K. CAM-K is expressed in really much more neurons, both glutamatergic and GABAergic neurons, during the embryogenesis. But later, they start to be expressed only in glutamatergic cells. So there is a switch of identity very often for CAM-K, for POMC, it is also true. So it might be that the same happens also with SIM1. That's why my question was, are there some SIM1 expressing cells during development that switch their epigenetic identity later to another one, so that they switch off the promoter of SIM1? So what we are looking at is not SIM1 neuron, but they also have the knockout of miR-7, because that's their setting. They do this starting from the embryonic state. That was my question. And if they don't discuss it at all, either in introduction or in discussion,
no,
they don't discuss that
No.
That their phenotype is actually embryonic, right? Do they?
No.
Yeah, I also didn't see that. So and that's we need to keep that in mind, because, as I said, I admire the work which has done there, and I really find it's really great paper. But we just need to keep it in mind that this phenotype is embryonic.
No words. Not at all. Oh, membrane. No. They don't have. They don't even have the word. Okay. So, then you asked me how many reads per sample have they had and how enriched was the library. So I couldn't find this info, and I even checked the supplementary data, and I couldn't find it. Maybe I didn't check well the supplementary data because also was a bit tricky, but I didn't see it.
No, overall they have quite a good expression, like based on this. Yes.
I don't really understand this question. How many reads?
Reads per sample. Yeah, so it depends on the depth of this of the sequence.
In here, right?
You mean in the sampling stage?
Yes. Yeah. So how deep is the sequencing? Because it depends on the sample, how many cells do you use, right? So how much of the tissue, and also how many reads do you use per sample, and from that you can know like whether like how deep was the sequencing. But here it looks like they had quite a decent number of reads for the samples, and the expression was pretty high.
Okay.
Where was this graph? Where's this graph? Yeah, yes, FPKM. So this is based on this figure 5C. That was what I was referring to. Yes, fragments per kilobase. Yes, per kilobase of transcript per million fragments. And like from here, it looks like a decent expression.
But I read some recently, they not always use the PKM to deal with their data. Now we always use CPM. It's another.
Yeah, but from here you can also at least see that looks this sequencing setting was okay. Yeah.
Okay. So, could you please provide more details about how do they analyze the data shown in Figure 6, this genome wax association, encode 5X phenom XCAN, and so. So this.
It's really cool, right? So when they associate different mutations with specific traits like height and so on, so I find it really interesting.
Yeah, so this one I'm not very familiar with about how these softwares work, but actually when I was analyzing the data now, because for example here, like in the other one, for example, they say that open targets reported a colocalization of these. Bueno, what type of diseases is linked to the disruption or the decreased expression of this protein, right?
In humans.
In humans, yeah, I assume yes, in humans. So one is like about the height, and then the other one they talk all the times about the trunk fat percentage.
That's about which gene?
About because the miR-7 is encoded in the 5 prime of this of this gene, right? So then they look for the disruptions, the consequences of the disruption or the decreased expression of this protein, and they claim several times that it is associated with increased height and trunk fat percentage.
So what is on the axis Y? Axis Y is log 10.
The association, no?
Or
Like
association. I mean, how do you measure association? Okay. So
Maybe-
it's highly associated. So the
the
highly
highest.
associated with standing height, right?
Yeah. So one is standing height, that is what they say. But about trunk fat, I couldn't find anything. I think impedance of arm and impedance right and left, impedance of whole body. I mean, like, of course, if you're impedant, maybe you have high trunk fat, but it doesn't mean that the trunk fat high.
What is on the x-axis? This is the genome location, or how is it, or what is this? Are those different different?
I think it's like
mutation points within this gene.
No,
from 5' to 3', or what is this?
What I understand is this is the type of diseases, nervous system disease.
Yeah, wait, but what? Well, somehow they have the X, so they stretch it to the.
Yeah, but I think it's just to make it fit. I don't think it's like.
I think so.
I don't think like the left, the right you go, the left you go, you change the value. I think it's just to make fit all the diseases.
Really? Like
Because-
it clusters somehow.
Because I think it's like, for example, infection diseases. Like, is there a high association of this a protein with the infectious diseases?
That's
It seems
maybe true, yes.
not really. And then, for example, this one is measurements, because it's not associated with a disease, just with a maybe blood measure. Also, they have uncategorized
Okay, maybe.
biological
yeah.
process, cardiovascular disease.
Maybe they just space them so that they fit several traits
Mhm.
per.
So you can see.
Oh, I see. So it's just one by one.
Yeah.
Now I got it. So there's just one per axis, and they just check all of them, have all of them aligned. Also, I see. Okay.
And yet they have this standing height that I would like to know what does it really mean.
No, standing height is like when you stand up and then you measure your height.
Yeah, just how tall you are.
Yes.
Okay. And then this impedance of arm that I think is what they link with trunk of fat, because is the following higher measure. And then there is.
I don't think so. It's probably from a different graph.
No, it should be here. It has to be here. It has to be here. And I check,
I also don't see.
and there is nothing related to fat itself, like deposits or the ratio of your waist with your thing. So this is very interesting.
So what does it mean that the lower means like the association? The lower the H on NR and PK, the lower the height. Correct?
The
I guess.
increased height, the less protein, this PK, the highest the height. The tallest the person.
Oh, okay. So it's kind of inverse. Inverse. I mean, those
Yeah.
triangle. If it points down, it means that it is. Then it is inverse correlation, I guess, right?
Ah, yeah.
Probably.
Makes sense.
But I don't see anything about
Fat.
trunk fat.
I think it's this. I think it's
No.
this. Because if you're impedant, think about it. If you're impedant on the whole body, your truncal fat percentage is going to be higher than a wall type phenotype. But if the increase of trunk fat is caused by the impedance.
Okay.
I don't know. Okay, and then so I had this question: is the lack of MicroRNA-7 causes a reduction of the S1 neurons in hypothalamus? How do they know that the phenotype is caused by the MicroRNA-7 role in metabolism or just by the neuronal loss? Because if MicroRNA-7 knockout causes neuronal loss, how do you know the consequences are metabolic or because of the neuronal loss?
Well, but this is still caused by the decrease of the by miR-7 loss. Even if it is because of the loss of SIM1 neurons, it's just the mechanism. It could be that miR-7 loss causes the embryonic not enough production of SIM1 neurons, which causes the phenotype which we see. Still, it will be
It still is
because
considered
of miR-7 loss, right?
Yeah.
Because they have a control, yeah.
Yeah, but the point is maybe MicroRNA-7 is not related to metabolism, but to cell survival.
Well, it will be anyway related to metabolism because if it causes the loss of SIM1 neurons, that also it is related to metabolism because SIM1 neurons are related to metabolism, so it decreases the function. I see what you mean. So it could be that the real target of miR-7 mutation in their case is the embryonic upregulation of some gene which causes this decrease of SIM1 neurons, and this per se causes all the phenotype. So, and they're looking for some genes like synuclein or some others in the adulthood, but these are already not relevant targets. The relevant target was in the embryogenesis, and that target was the main, or several targets were the main ones which caused this this decrease of CM1 neuron number. Which percent causes all these
Exactly.
These phenotypes which we see? Yeah, probably it is like that. Most probably this is exactly like that. Can you get back, please?
Yeah.
So from here it is just about this gene. So and they use it using which tool? Which tool?
It's this thing. Yeah, sorry. EQTL.
EQTL. This is the tool. This is
Yeah, I think that's a tool plus open targets. I think
open targets.
yeah, which is I think it's open targets a database, and I think
Okay.
the EQTL is the
And-
software.
But which tool do they use to associate a specific point mutation in this GWAS? Yeah, so the genome-wide association studies with the specific traits. If you go back,
Yeah. Right.
you see here they have some. You see here they have the genome. On the bottom, on the very bottom, they have the genome. And then from here, they infer that they go further, like zoom in, and then they show the specific loci and then association with some, you see standing height,
Impedance of whole body.
right, trunk fat,
Here it
trunk
is.
fat ratio, all of this. Triglycerides. It's very, like, this one is very interesting for me. So this looks really cool. So how did they do that?
Like I think what I understood is that there is this database available of clinical cases that for some reason the genome is reported or some sequencing is done, and they have found these changes or these mutations or this decrease of the protein because of
My question
genetic
is like
changes.
which database did they use to correlate those specific point mutations on the genome scale with these different that on the bottom is very interesting, yeah?
I think it's open targets, if I'm not wrong. But I didn't look further
But you
into that.
told about some phenome X scan, some
Yeah.
5X, some the encode. So you didn't look into it, right?
Yeah, maybe it's from here as well.
Interesting.
We can check in the paper.
So which three? Okay, it's okay. We don't need to check now. So the three graphs they use here is, you see they align relative age of first hair.
First fashion hair.
Yes, they have especially relevant for female, probably. Standing height GWAS and then EQTL.
I think they just check what is related to this mutation on this
I'm
gene,
just trying to figure out how do they do this.
and they just
It's
found.
beautiful. I really like it.
Yeah, but it looks a bit random to me.
I mean, this is mutation. Mutations are random. Yeah. And
What I mean is not really relevant to a pathologic phenotype. More like
it is. I mean, this is the way they link it, and they claim that could be also the reason in humans. And like for me, it looks pretty convincing. Yeah. So they have height, they have fat-related triglycerides, and this all kind of fits to what they measure.
Yeah.
Of course, we know that it's developmental, but it could be also developmental in humans.
It's true.
So it could be that this exactly the same thing happens there, and since miR-7 is so conserved, that I would not be surprised. So it looks really good. You know,
I'm surprised, however, that when I check where is the maximum expression of a miR-7, they said it was in kidney, but they are not diseases associated or at all with kidney.
Wow. miR-16 is, for example, also one of the highest expressed miR-RNAs in the brain. Yeah, and also in the hypothalamus.
No, miR-7.
No, I
right?
know. They studied miR-7 before. This particular group, they studied it in the pancreas, and
Oh, yeah.
they already
It's true.
used to recommend that.
Okay.
And I just want to check. So that's really beautiful. Yeah. And which graph you just showed before about the kidney? Where is it?
This is I searched it myself from the Protein Atlas and UniProt.
So did you find something?
Pues
About system one?
in UniProt they say that these microRNA might have effects during embryogenesis, might have. So I don't know what does it mean. And then this is the consensus data set about the RNA tissue specificity is mainly kidney, hypothalamus. That makes sense, right? Pancreas also makes sense.
Wait, this is for which?
miR-7. miR-7, yeah.
In humans?
In human.
Okay.
Yeah, hypothalamus, pancreas, adipose tissue, that all makes sense. Also, they split midbrain, thalamus, amygdala, so it's all kind of...
That's in the adult?
I assume yes, because I don't think they could measure in other states, right?
We still have so much that we don't know, you know?
Yeah.
You know?
And we cannot know. I mean, legally wise.
That's why we are doing research. We are trying to I mean that some questions were not even asked. You know?
So okay, shall we move on?
Sure.
So this is the other paper that you mentioned, the miR-7 and miR-1792 in the Arcuate nucleus of mouse hypothalamus regulates sex-specific diet-induced obesity.
It was published 3 years before that, right?
Yeah, 19. This is very new, 22. Yeah. So I did this table more or less comparing both of them. So
that's cool.
The target, in the second paper, they analyzed the same target and also a different one. So they both, the first one does a total miR-RNA 7 knockout. The second one focus on this miR-RNA 7A1. They do knockouts each one by one, and then they analyze this other cluster.
Separately.
Yeah, they analyze them separately, and then they decide to go for 7A1 because it was the highest. It was the one with highest expression.
I don't think they knock out. They knock down.
No, I'm gonna explain now what the knockout they do. So then they also did the miR-1792 cluster, and they focus on the 92 as well. The same reason, because it's the one with highest expression.
And which group? Can you remember?
So
POMC, I think. POMC, that's what they do, right?
So the thing is that.
No, I mean for 1792.
Yeah, I think it's the same.
And 7A1.
I maybe forgot to write it. So for the knockout, for the first one it's pre-mediated excision, but for the second one they use miR-RNA sponge. So it's not embryonic state; it's in adult state. It's something that they supply.
Exactly.
They supply a miR-RNA
so this
sponge.
Is adult. So in this case, it's adult phenotype. And the same also
I think
they use for that one as well, right?
I think so.
As well, right?
I think I forgot to write it. I think it's the same.
And it is also focusing on a different neuronal lineage, right?
Yeah.
It's POMC. In case of Stoffel's paper.
The first one?
Certainly the first one.
Uh-huh.
They I think they check POMC.
They check
And they see
POMC.
some slight. What remind us? What do they see?
It was like minor change.